In the silken hush of the Archean world, long before the breath of animals or the bones of plants, something stirs not with limbs or light, but with chemistry. The early earth, still cooling from its fiery youth, exhales a veil of volcanic gases into an atmosphere untouched by oxygen. Methane shimmers through the haze. Hydrogen seeps through fractured crust. Rain falls warm and mineral-rich into alkaline seas. And within these quiet waters, unseen by any eye that will ever blink, the earliest bacteria begin to shape themselves, not with intention, but through the slow arithmetic of entropy and energy. No nucleus shelters their genes. No wall yet defines their edge. They are forms, barely formed membranes drawn from fatty molecules enclosing strands of replicating code. They drift. They absorb. They divide, though not perfectly. Mutations ripple gently through each lineage, soft deviations without direction. In these first microbial shadows, there is no struggle, only reaction. Molecules pass from outside to in. Energy flows from reduced compounds to oxidized forms. It is metabolism without memory function, without face. These early cells are not oxygen breathers, nor yet builders of structure. They rely on the chemistry around them. Hydrogen, sulfide, ferrous, iron, carbon dioxide, simple substrates to drive proton gradients across rudimentary membranes. ATP forms in tiny pulses. Life, such as it is, continues. There are no predators here, no competition, only quiet occupation. Rock, water, and gas offer all the world there is, and life flows into it like ink into cloth. Some cells drift into the shade of hydrothermal mounds. Others cling to mineral particles, exchanging electrons in silence. The first bacterial lineages do not yet have names nor distinctions we can trace. Their differences are metabolic, genomic, hidden in the sequences that will one day fossilize in databases, but which now evolve like breath on glass. Only fragments of this world survive. Isotopic signatures in 3.8 billion-year-old rocks from Greenland. Carbon ratios in ancient graphite. The smallest hint that something once lived and metabolized in a sea without oxygen or sunlight. These traces do not tell stories. They do not cry out. They remain fixed in crystalline lattice, quiet as memory. And yet from this vague genesis, an entire domain of life begins to unfold. The bacterial domain, vast, diverse, structurally simple and biochemically profound, finds its origin not in a single spark, but in a slow murmur of chemistry becoming persistence. No fossil marks the first cell. No clear ancestor unites them all. Perhaps there were many origins, competing protocells, gene-swapping membranes, chemical possibilities, that flickered and went dark. What remains is not certainty but continuity. Modern bacteria carry echoes of these earliest forms in their genes, in their enzymes, in the fragile universality of the ribosome. It is almost comical in a long-sleep sort of way how little separates the most primitive cell from the bacteria of today. A membrane, a loop of DNA, a set of ribosomes that's enough. Billions of years later, these are still the fundamental tools of life. All else, the shapes, the behaviors, the adaptations are refinements on this minimal theme. In this beginning, time does not rush. 1,000 years pass between useful mutations. 10,000 more before a new enzyme arises. No one is watching. No records are kept. But in the stillness, cells divide. Errors accumulate, and with them the possibility of novelty. Genes that help gather iron or stabilize membranes, or exploit a new energy source, they persist. Others fade. The cell walls thicken. The membranes stabilize. Some lineages begin to specialize to favor certain compounds or habitats. The sea becomes a mosaic of niches, vent water, shallow pools, rock surface, anoxic mud. Still, no one competes. Life is not yet fast enough for conflict. We may never know what these first bacterial lineages were called or whether we would recognize them as distinct. The fossil record when it begins is not of cells, but of their habitats, rippled sediments, precipitated minerals, subtle isotopic imbalances. The biosignatures are indirect and interpretation is always cautious. Perhaps it is fitting that the beginning of life leaves no body behind. Only chemistry can testify, and chemistry does not speak, and so we rely on inference, on molecular clocks and ancestral reconstructions, on comparative genomics where genes from modern organisms are aligned and unwound, their histories traced backward through branching trees that do not end in roots but dissolve into fog. We map the present in hopes of glimpsing the past, but there is no consensus about the shape of the earliest tree. Some argue for a thermophilic ancestor born near heat and sulfur. Others suggest a photosynthetic lineage tracing back through light-harvesting pigments. Still others wonder if early life was communal, a gene-sharing swarm rather than discrete lineages. Here even classification becomes paradoxical. We seek to name what never named itself, to draw boundaries around forms that lived before formality, to assign lineages to cells that blurred and merged and shared as freely as the molecules around them. Bacteria by their very nature resist the simplicity of lineage. Their genes hop, their identities shift. The deeper we look, the more structure dissolves, and yet something persists. Through all this drift and uncertainty, something bacterial emerges not as a shape but as a rhythm, a way of being alive that is elemental. Absorb, divide, persist. There is elegance in that simplicity, not crude but refined. Like the first shimmer of code in a sleeping computer, a self-executing loop patient and recursive waiting not to be seen but only to endure. The first bacteria do not seek complexity. They do not innovate by design, but over time they elaborate. Small refinements accumulate new transporters, regulatory loops, protective enzymes. A lineage becomes better at one thing and then another. What began as a generalist becomes a specialist. And so evolution proceeds not as a ladder but as a spreading stain, branching, fading, overlapping. Most lines vanish, a few persist, and from those few the full spectrum of bacterial life will unfurl. Here at the edge of the beginning nothing is clear, not form, not function, not lineage, only the fact of persistence, that in a world without eyes, without lungs, without words something continues. It divides, it adapts, it remembers not in mind but in code. And through that memory it changes very slowly, beneath the slow tides of Archean shorelines long before soil or sand colonies of bacteria begin to settle. Not individuals but collective cells gathering in thin, pliant sheets that ripple with metabolism. These early mats do not float freely. They cling to stone, to silt, to submerged outcrops warmed by the sun. Layer upon layer microbial films grow, each stratum built from excreted polysaccharides, secretions that hold cells in place and invite others to join. They are sticky, flexible, and patient. Over time, sediment falls into these mats and is trapped. Light filters through the upper layers, fading as it passes into the deeper ones. A vertical world of function begins to take shape. Above the phototrophs, below the anaerobes, in between a quiet spectrum of microbial roles, each defined by what little light or compound remains. Stromatolites begin to form not as planned structures, but as accidental monuments to persistence. Millimetre by millimetre, the mats build domes and cones, bulges and ridges shaped by the tides and the wind. No one directs them. No blueprint guides the work. It is life as architecture built from time and repetition. Some of these stromatolites will fossilize, surviving 3 billion years or more, quiet mounds in ancient carbonate rock. Others will be worn away, their builders long forgotten, their forms softened back into sediment. But in these structures lies the first visible trace of life. Long before shells or spines, before trees or bones, there is this thin films of microbial exudate layered and laminated, the slow breath of colonies turned to stone. These mats are not inert. They teem with activity too small to see gradients of oxygen, sulfur, nitrogen, and iron, redox reactions cascading through the strata. The cells communicate chemically, release enzymes, build quorum. Though they lack nerves or synapses, they coordinate. A chemical gradient is enough. A signal molecule diffused from one side to another can shift gene expression, regulate growth, guide the formation of new filaments. Here in these mats, bacterial life becomes more than a single cell struggle. It becomes community, shared space, shared metabolism, and in a sense shared memory. The mat persists even when individual cells die. The structure remains. The biofilm survives as an ecological scaffold inhabited again and again by generations of cells that inherit not only genes but placement and architecture passed down, not by instruction, but by presence. Even today, microbial mats remain in hypersaline lagoons, hot springs and oxygen-poor basins. They're ancient but not extinct. In Shark Bay, Australia, modern cyanobacterial mats still form stromatolites, much like those that once flourished 3 billion years ago. The same slow-building pattern, the same blue-green tinge, the same sticky threads of exopolysaccharide. It is a quiet defiance of time what worked, then still works now. Evolution has refined but not replaced them. The irony lingers that one of the oldest living ecosystems is also one of the simplest and one that modern multicellular life rarely even notices. And yet their survival raises questions. Why do microbial mats thrive in extremes in salt-heat desiccation? Perhaps because they were born in such places. The early Earth was no gentle garden. Ultraviolet radiation bathed the surface. Oxygen was toxic to many cells. Salinity and temperature fluctuated wildly. In that context, mats were not relics but adaptations. They provided shade structure microenvironments. They created internal gradients where cells could retreat into safer chemistries. They were microbial cities before cities had names. Still much remains uncertain. How did these communities emerge from free-living cells? Did mats evolve once or many times? Are modern stromatolite builders direct descendants of their Archean counterparts, or simply inheritors of an ecological role the fossil record cannot say? Molecular clocks offer estimates, but the genes involved in mat formation are not always conserved. Horizontal gene transfer muddies the waters. One lineage may borrow a useful trait, spread it among others, and disappear, leaving behind a function divorced from ancestry. These questions are not just academic. They touch on how we define life, lineage, and survival. If a community endures but its members change, is it the same? If the structure remains but the genes differ, does the lineage continue? Such ambiguities are part of microbial evolution. Lineage in bacteria is not a simple tree. It is a network woven from threads that cross and loop, tangle and merge. Cells divide, yes, but they also exchange genes, plasmids, mobile elements. The mat as a whole can acquire a new function through the arrival of just one novel bacterium, a subtle shift that ripples through the community. In the quiet heat of these ancient shallows, even such ripples take time. A new photopigment might spread over centuries. A mutation in exopolysaccharide production might take millennia to reshape the mat's surface, but time is abundant. In this microbial epoch there are no seasons, no rush. Change is measured not by generations but by geochemistry. And yet, for all its stillness, the mat is dynamic. It responds. When light shifts, upper layers bloom with phototrophs. When oxygen builds, anaerobes retreat. When tides bring new minerals, reducers and oxidizers awaken. It is a choreography of chemistry enacted by organisms too small to see, too slow to follow. And still it continues a persistence that outlasts extinctions, tectonic shifts, even continents. These mats, for all their simplicity, represent a turning point. Before them, life was diffuse individual floating. With them, life becomes spatial, structured, ecological. It begins to shape its environment, not just adapt to it. The mat modifies the chemistry around it, alters sedimentation, precipitates minerals. It is not yet engineering, but it is something close. The earliest example of life as landscape. Modern analogues suggest similar patterns. In Yellowstone's hot springs, colourful bands of microbes arrange themselves by temperature tolerance and light availability, natural zoning along thermal gradients. In hypersaline ponds, different microbial pigments tint the water, red, green or violet. These patterns are not aesthetic. They are spatial signatures of ancient processes still playing out in microbial time. Somewhere in these patterns lies a mystery. Why in some places do mats grow thick and fossilize while in others they dissolve or decay? What determines their preservation? Is it luck, mineral saturation, microbial composition, or simply time? The fossil stromatolites we study are the survivors, the exceptions. Most mats left no trace. Their builders lived layered and vanished, swept away by currents or consumed by other microbes. It is a silent bias in our record, the visible favouring the durable. The delicate stories are lost. Still, the layered stones speak. In Pilbara, in Warrawoona, in Isua, the old rocks carry whispers of microbial labour. And though we cannot name the species or sequence their DNA, we know their signature. The domed rise, the laminated structure, the chemical gradients etched into carbonate. It is enough to say life was here, not briefly, not rarely, but abundantly, and it built. Long before inheritance becomes vertical, before descent locks lineages into familial chains, bacteria begin to discover another way of sharing. Not the slow passing of genes from parent to offspring, but the abrupt, unceremonious exchange of code between strangers. Lateral gene transfer, as it will come to be known, is not invention but diffusion, a microbial whisper passed cell to cell carried on plasmids, transposons, viral capsids. Genes once confined to one lineage slip into another, bringing with them enzymes, resistance, entire metabolic capabilities. A bacterium that once digested sugar might overnight acquire the machinery for nitrogen fixation or arsenic reduction. The genome once thought to be a static script reveals itself instead as modular, adaptable, permeable. In this bacterial world, identity is not a closed loop but a fluid mosaic, traits assembled not only by mutation but by contact. The implications are quiet but immense. Evolution does not wait for slow branching. It recombines, it shortcuts, it copies and pastes. Across oceans, across phyla, across barriers we once assumed to be impassable. A bacterium near a hydrothermal vent might share genes with one in a tidal lagoon. Not through intention, but through chance, a virus that infects both a shared space on a migrating particle. A brief fusion of membranes. What begins as accident becomes strategy. Some bacteria develop competence, the ability to uptake naked DNA from the environment. Others evolve conjugative, peely tiny bridges through which they pass plasmids. And still others, like bacteriophages, act as couriers of genetic fragments, infecting one host, picking up a gene, and delivering it to the next. It is not chaotic, but it is unpredictable. A gene's history no longer follows a clean path. It loops, detours, crosses. Like a stone skipping across water, its presence in different lineages leaves patterns but not origins. This lateral sharing renders bacterial evolution profoundly different from that of multicellular life. In plants and animals, lineages diverge with time accumulating differences. In bacteria, lineages blur, overlap, and sometimes converge. A thermophile in an acidic spring may have acquired heat resistance genes from an entirely unrelated species. A pathogen may borrow a virulence factor from a soil microbe. These transfers are not rare exceptions, they're frequent, ongoing, essential. In fact, they are responsible for some of the most striking features of bacterial biology: the spread of antibiotic resistance, the emergence of new metabolic pathways, the rapid adaptation to changing environments. To imagine bacterial genomes as static is to miss their essence. They are not scrolls but patchworks, not preserved texts but living documents constantly revised. And in this revision there is no editor, no oversight, only fitness persistence and biochemical compatibility. This process complicates classification. If genes can cross boundaries, how do we define a species by shape, by habitat, by a core set of conserved genes? Even these markers become unstable under the pressure of horizontal transfer. Two bacteria may share over 90% of their genes, and yet belong to entirely different ecological roles. Others, nearly identical in function, may diverge widely in sequence. The tidy categories of classical taxonomy genus-family order dissolve when confronted with this genomic fluidity. What remains is a kind of probabilistic clustering species defined not by strict boundaries, but by centers of genetic coherence. Around these centers swirl accessory genes, mobile elements, variable islands of function. It is less a tree and more a cloud, an outline sketched from averages and exceptions. Still we persist in classifying. The human mind craves order even when nature refuses to offer it. We construct phylogenetic trees not as maps of certainty but as tools of approximation. These trees built from conserved genes like 16S rRNA offer a glimpse into deep relationships, but even they are not immune to horizontal disruption. A single transferred gene can skew the branching, alter the inferred ancestry. The trees shift depending on which genes are chosen, which methods applied. There is no perfect resolution, only better models, more complete data, and a growing acceptance that in the bacterial world lineage is always part fiction. And yet even fiction can reveal truths. The pattern of gene transfer is not random. Some genes resist movement, those deeply integrated into core cellular processes like transcription or translation. Others move freely, those involved in surface structures, environmental sensing, specialized metabolism. This bias reveals an evolutionary economy, core stability, surrounded by peripheral innovation. The bacterial cell protects its heart while adapting its edges. It is a strategy both conservative and flexible, allowing for rapid shifts without destabilizing essential systems. This architecture, a conserved core genome and a flexible accessory genome, becomes a defining feature of bacterial species. It explains their resilience, their adaptability, their ubiquity. There is a soft irony here, that the simplest organisms have the most complex evolutionary dynamics, that cells without a nucleus or multicellular organization can outmaneuver more advanced life forms in genomic plasticity, while animals spend millions of years refining traits through reproduction. Bacteria can acquire the same function in a single gene transfer. Complexity, it seems, does not always correlate with power. In this microbial world, simplicity becomes a kind of strength. This also challenges our understanding of ancestry. If genes move across lineages, then what does it mean to trace a bacterial lineage? Are we tracing cells or functions? Are we following the history of organisms or the history of genes? Often the two do not align. A gene may have one origin, its host cell another. The modern bacterium is a chimera, a collection of borrowed, inherited and modified parts. And yet it functions, it persists, it thrives. One of the great mysteries that emerges here is the origin of complexity itself. Did early life use lateral transfer more than it does today? Was the last universal common ancestor, if it can even be said to exist, a single cell or a community of gene-sharing protocells? Some propose that early evolution was not Darwinian but communal, a pre-Darwinian era of rampant exchange only later constrained by cellular lineages. If so, then individuality itself may be a late invention. For a time, life may have existed as a network, not an organism, a shared experiment in metabolism and replication. Such speculations are grounded in data, but they remain speculative. The deep past offers few clues, and those that remain are obscured by the very process we seek to understand. Horizontal gene transfer erases its tracks. It leaves behind function, not provenance, and yet its impact is clear. It is why bacteria adapt so quickly, why they appear in every environment, why classification remains elusive. It is the background hum of microbial evolution, quiet, constant, and profoundly influential. In the end, perhaps the most poetic aspect of lateral gene transfer is its indifference. It does not favor one lineage over another. It does not follow rules of kinship or descent. It simply occurs where conditions allow, where compatibility permits. A gene floats, finds a host, and continues. There is no plan, only persistence. And through this microbial promiscuity, the bacterial world continues to diversify, to adapt, to endure. When iron-rich seas shimmer under the ancient sky, long before oxygen comes to claim the atmosphere, another lineage begins to rise, not through conflict or conquest, but through quiet appetite. These are the iron breathers, bacteria that feed not on light or sugar, but on the oxidation of ferrous iron. In the reducing environment of early earth, iron is soluble, abundant everywhere, and where there is chemical potential, life finds a way to harness it. These bacteria do not swim or hunt. They dwell on mineral surfaces, forming biofilms along the ocean floor, etched into the crevices of basaltic rock lining hydrothermal seeps where warm water leaks from fractured crust. Here they oxidize iron, converting Fe2+ into Fe3+, and in doing so generate energy just enough to fix carbon to grow to divide. The byproduct of this metabolism, ferric iron, precipitates out of solution, forming insoluble oxides. Layer upon layer, this rust-coloured sediment settles, creating what will one day be known as banded iron formations. They are not deposited in haste. Each stripe, each layer records a slow rhythm of microbial metabolism and geochemical change laid down over tens of millions of years. Some bands are thin, delicate traces, others meters thick stretch for kilometers underground. They are the geological footprints of ancient biochemistry fossils, not of bodies, but of processes. What these bacteria lack in visibility, they compensate for in impact. Nearly all the iron mined today steals ancestor blood's essential core originates from their quiet labour. That the modern world is shaped by ancient microbes without form or face is one of evolution's subtler ironies. These bacteria lived in darkness beneath shallow seas with no audience, no awareness. Yet their metabolism altered the ocean's chemistry, changed the redox state of the planet, and left behind monuments of mineral that would one day define continents and economies. There is dry humour in this, the idea that rust, the most mundane of oxidations, was once a signal of life's slow expansion. Not all iron breathers are the same. Some use oxygen in trace amounts to drive their oxidation. Others rely on nitrate or even light. Photoferrotrophs, for example, perform an oxygenic photosynthesis, using light to oxidize iron instead of water. These bacteria may have played a role in both iron deposition and the early photic ecosystems of the Archean Ocean. Their pigments differ from those of modern cyanobacteria. They use bacteria chlorophyll absorbing wavelengths where light is faint, filtering through turbid water. In this dim spectrum, evolution finds yet another strategy. Energy is not abundant, but it is enough. Just enough to fix carbon, just enough to persist. The diversity of iron-oxidizing bacteria reflects a fundamental truth. Metabolism evolves not through invention, but through adaptation. Life does not wait for perfect conditions. It uses what is available: iron, sulfur, hydrogen, light, carbon dioxide, and slowly shapes biochemistry around it. Some lineages become specialists, others generalists. Some fade into extinction, others persist, but all are bound by the same imperative to extract usable energy from the surrounding chaos. In this sense, metabolism is the first logic of life, a negotiation between thermodynamics and opportunity. Despite their geological legacy, iron-oxidizing bacteria remain poorly understood. Many are unculturable, their genomes only recently sequenced through metagenomic assembly. Others are known only from environmental gradients layers of red and orange, where water meets rock, faint electrical potentials measured at the interfaces. Their metabolic pathways, while inferred, are often incomplete. Key enzymes remain unidentified. Regulatory systems are cryptic. The microbial mats they form in modern hydrothermal vents are ephemeral, shifting with temperature flow rate and mineral availability. And in the ancient record, direct evidence is always indirect, a challenge of interpreting chemistry, as biology of finding life in the residue of stone. Even the formation of banded iron layers is not fully settled. Some propose abiotic oxidation, UV-driven reactions, or photochemical processes. Others argue for a combined model: biology initiates oxidation, geology preserves it. The rhythm of the bands, alternating red and grey, suggests periodicity, perhaps linked to seasonal changes, volcanic pulses, or microbial succession. But the true causes remain open, unresolved. As with so much in early Earth science, what we know is overshadowed by what we suspect, and even more by what we cannot know. There is a quiet satisfaction in this uncertainty, that even processes responsible for billion-ton ore deposits resist complete explanation, that life's most enduring traces are not always the most interpretable. The fossil record is rich in silence, and the bacterial world with its minimal form and maximal effect thrives in that ambiguity. Today, iron-oxidizing bacteria still exist, though they rarely dominate. They line the interiors of iron pipes, corrode underwater infrastructure, flourish in acidic mine drainage. Their habitats are extreme, but their strategies remain ancient. They are not relics, they are survivors. And they remind us that evolution is not progress but endurance, that some forms refined early never need to change. In this way, the story of iron breathers intersects with a broader theme in bacterial evolution, that many innovations occur early and persist, not because they are superior, but because they are sufficient. Ferrous iron abundant in the Archean Sea provided a reliable electron donor. Oxidizing it required only modest enzymatic machinery. The yields were low, but the costs were lower. It was a viable path, slow, steady, survivable. And in evolution that is often enough. The presence of such metabolisms in modern extremophils, acidophilic iron oxidizers in hot springs for example, raises another quiet question. Are these lineages ancient holdovers or modern convergences? Have they survived in isolation since the Archean? Or have different bacteria rediscovered iron oxidation independently multiple times? Across evolutionary history, genomic data is inconclusive. The relevant genes are often found on mobile elements or embedded within atypical genomic contexts. Phylogenies of key proteins show both deep branching and lateral scatter. The truth may lie somewhere between ancient pathways preserved, but also reacquired, re-engineered, recombined. What remains clear is the centrality of iron to life's deep past. Not just as a nutrient, but as a scaffold for metabolism, a medium for electron flow, a canvas for geological art. The rusted landscapes of the early Earth are not mere background. They are microbial portraits formed not by brush but by breath. And in those breathings, slow and invisible, we find the first traces of planetary change. That such tiny organisms could shift the chemistry of oceans, precipitate vast mineral beds, and leave a legacy measured in tectonic strata is one of Earth's quiet paradoxes. That they did so without awareness, without design, only deepens the irony. Life in its simplest forms becomes geologic force. Evolution at its quietest becomes world-shaping. And in the flicker of redox as iron yields to rust, the microbial world takes its place not beneath geology but within it. Amid the quiet diffusion of gases and the rusted minerals of ancient seas, a new lineage of bacteria begins to alter the planet in a way no other has. Cyanobacteria, often called blue-green algae, though they are neither algae nor always blue, emerge with a quiet innovation that will change everything, not through conquest or complexity, but through the slow, persistent release of oxygen. They are among the first organisms to perform oxygenic photosynthesis, using water as an electron donor and sunlight as the energy source to fix carbon dioxide into organic matter. In doing so, they release molecular oxygen as a waste product, a gas that until now has had no real presence in Earth's atmosphere or oceans. The reaction seems unremarkable at first light, water and carbon dioxide yielding sugar and oxygen, but the implications stretched across geological time are vast. Cyanobacteria do not seek transformation. They simply metabolize. They absorb photons with their specialized pigments, chlorophyll, phycobilins, anchored in internal membranes. They live in colonies, filaments, solitary cells. They drift in the photic zones of shallow waters, settle into microbial mats, occupy niches others cannot, and slowly, molecule by molecule, they begin to exhale the future. At first, this oxygen does not accumulate. It reacts with iron, forming insoluble oxides that settle into sediments. It binds with volcanic gases, forming acids. It is chemical novelty in a world of reducers. But cyanobacteria persist untroubled by the fate of their byproduct. They continue to divide, to photosynthesize, to generate oxygen in excess of consumption. Over millions of years, these small emissions begin to outpace the planet's buffering capacity. Oxygen levels rise first in localized waters, then more broadly, eventually touching the atmosphere. The Great Oxygenation Event, as it will later be known, does not occur all at once. It takes hundreds of millions of years. It is not dramatic. There is no bang, no visible shift. Just the slow suffocation of anaerobic life, the oxidation of surface rocks, the transformation of Earth's redox balance. Many existing bacteria cannot tolerate the new conditions. Oxygen is toxic to cells unequipped with protective enzymes. Superoxide radicals damage DNA, proteins, lipids. For vast microbial populations, this rising gas spells extinction. Entire lineages vanish, leaving behind no fossils, only gaps in metabolic inheritance. The cyanobacteria remain. They are not immune to oxygen's toxicity, but they adapt. They evolve catalyses, peroxidases, superoxide dismutases, enzymes that neutralize reactive oxygen species. Their membranes adjust. Their internal systems reorganize. They continue to thrive in the shallow waters where light is abundant. Some become filamentous, forming long chains with specialized cells. Others evolve heterocysts to fix nitrogen under oxygen-rich conditions. Their innovation becomes a platform for further innovation. In some microbial mats, they dominate the uppermost layers, shading and shaping the communities below. In others, they form blooms, expanses of pigmentation that ripple across calm lagoons. Their pigments, deep blues, vivid greens, soft reds, are not decorative. They are tuned to specific wavelengths optimized to harvest energy from a sun filtered by water and atmosphere. There is quiet elegance in this efficiency, no excess, no waste, only what is needed. The fossil record begins to reflect their presence more clearly. Stromatolites formed by cyanobacteria exhibit distinctive lamination preserved in carbonates for billions of years. These structures still dot the geological record, though their interpretation is cautious. Abiotic processes can mimic biotic ones, but when paired with isotopic signatures, light carbon isotopes indicating biological fixation, the evidence strengthens. Cyanobacteria are not just survivors, they are architects of change, and yet their origin remains a mystery. When did oxygenic photosynthesis evolve, from which ancestral lineage? The photosynthetic apparatus is complex photosystem I and two chlorophyll-binding proteins, water-splitting complexes. These systems suggest a long period of refinement, perhaps predating the cyanobacteria themselves. Some hypothesize that horizontal gene transfer played a role, assembling photosynthetic pathways from disparate ancestors. Others argue for a deep singular origin, with cyanobacteria refining and stabilizing the process. Genomic data provide fragments of insight. The core genes of photosynthesis cluster in cyanobacteria, but share homology with genes in other phototrophs. The sequence divergence is too great for clarity. The molecular clock ticks softly, obscured by ancient transfers and deep time. There is also uncertainty about the environmental context of their rise. Did cyanobacteria first emerge in freshwater pools protected from saline stress? Or did they evolve in marine environments where light and nutrients were more consistent? Modern representatives are found in both. Some prefer quiet lakes, others drift in the open ocean, forming vast populations in the euphotic zone. Still others colonize desert crusts, thermal springs, polar lakes. Their ubiquity today offers little help in locating their origin. Like many bacterial lineages, they have expanded beyond the bounds of inference. What is clear is their impact. Oxygen accumulation reshapes Earth's geochemistry. Manganese and sulfur cycles shift. Methane, once a dominant greenhouse gas, is oxidized, triggering global cooling events. Some researchers link the rise of oxygen to the Paleoproterozoic glaciations, planetary-scale feedbacks initiated by microbial metabolism. Life in its quiet persistence alters the climate, the oceans, the very air. And with oxygen in the atmosphere, new possibilities emerge: aerobic respiration more efficient than fermentation, ozone formation protecting surface life from ultraviolet radiation, the eventual rise of complex multicellular organisms. All of this flows from a bacterial lineage, from light harvesting pigments, from water split in photochemical chambers. Cyanobacteria, for their part, do not change dramatically. Their forms remain consistent across eons. Their genomes are large by bacterial standards, with many regulatory elements and secondary pathways. Some produce toxins, microcystins, anatoxins, perhaps as defense, perhaps as metabolic byproducts. These compounds affect modern ecosystems, but are likely ancient, predating higher organisms. The bacterial world does not wait for observers to justify its complexity. It acts, adapts, survives. Today, cyanobacteria are studied intensely, not just for their evolutionary role, but for their ecological impact. They contribute significantly to global carbon fixation and nitrogen cycling. In oceans, genera like Prochlorococcus and Synechococcus dominate microbial populations. Despite their simplicity, they are essential components of planetary function. Genetic studies continue to uncover their diversity, their adaptability, their role in shaping the biosphere. They are used in bioengineering, considered for terraforming, explored for biofuel production. and yet they remain what they have always been: bacterial cells, harvesting light, exhaling oxygen, building worlds without ever intending to. That the breath we take is rooted in their waste, that the ozone we rely on is spun from their exhalation, that the planet itself is no longer as it was before their rise. These are facts too vast to comprehend fully. And yet, they are true. The blue-green shimmer at the edge of a pond, the haze of a bloom beneath a summer sun, the thin laminae of carbonate stone. These are the quiet residues of a bacterial lineage that never sought to dominate, only to persist. And in persisting reshaped the world. It arrives without sound, without witness, without warning. The great oxygenation event unfolds not as a catastrophe in the human sense, but as a slow, inexorable chemical shift, one molecule at a time, one breathless lineage at a time. For hundreds of millions of years, cyanobacteria continue to exhale oxygen, not with ambition, but with biochemical necessity. And as the gas accumulates, it does not elevate or enlighten. It poisons. To anaerobic life, once the dominant form across oceans and sediments, oxygen is not a gift. It is a reactive, destabilizing force, an agent of cellular destruction. Enzymes that once operated freely now misfold. Membranes oxidize. Metabolic pathways collapse. The ambient chemistry that sustained early life begins to vanish. Molecules once soluble become toxic. Reduced metals precipitate out of solution. Sulfide, once abundant, declines. Whole ecological systems, invisible and unrecorded, erode under this new atmospheric regime. Bacterial lineages that had thrived in anoxic comfort for over 1 billion years begin to disappear quietly without fossil or eulogy. There is no mass extinction layer, no crater, no soot. Only gaps. Genomic silence where once there was code. The irony is that these extinctions are driven by life itself. The oxygen is not volcanic or meteoritic, it is biological. Its source is photosynthesis, its cause the simple need for energy. Cyanobacteria do not intend harm. They are indifferent. They persist, divide, and continue to metabolize. But in doing so, they render the world hostile to the very communities from which they emerged. It is perhaps the earliest instance of ecosystem collapse driven by an internal feedback loop, life altering its environment so profoundly that it becomes toxic to itself. Yet even this is not total. In the sediments, in the cracks of stone, in deep marine basins, anaerobic bacteria find refuge. They shrink into marginal habitats, adapt to low oxygen. Niches evolve mechanisms to detoxify or avoid. Some form spores, others retreat into biofilms. A few develop ingenious metabolic workarounds using nitrate or sulfate in place of oxygen. And in these refuges they endure. Evolution does not demand dominance, only continuation. Meanwhile, oxygen begins to accumulate in the atmosphere. At first levels remain low, but over millions of years a tipping point is reached. Reactive gases such as methane are oxidized and the greenhouse effect weakens. A global cooling begins, glaciers advance. The Paleoproterozoic ice ages arrive, possibly triggered by the very oxygen that now drifts upward from ocean to sky. This interplay between microbial metabolism and planetary climate marks the dawn of Earth's modern atmospheric system. The sky, once tinted with methane and haze, begins to turn blue. The ozone layer forms, shielding the surface from ultraviolet radiation. New metabolic possibilities emerge. Aerobic respiration, a far more efficient way to harvest energy. The potential for complexity increases not because life reaches upward, but because the chemistry now allows it. Yet the transition is far from smooth. Oxygen is not evenly distributed. It forms local oases, thin layers near the surface pockets within microbial mats. Stratified waters develop with oxygenic zones above and anoxic depths below. Bacterial life responds accordingly. Aerobes evolve and rise. Anaerobes retreat or adapt. The redox gradients become more complex, more layered. Microbial communities restructure around these chemical landscapes. Some bacteria learn to use oxygen directly. Others scavenge reactive oxygen species or exploit the gradients between oxidized and reduced environments to drive novel metabolisms. This period is one of both loss and innovation. Extinction clears the canvas. New lineages sketch themselves onto the oxygenated world. The fossil record remains sparse, but chemical evidence grows. Chromium isotopes, molybdenum levels, sulfur fractionation all suggest a planetary shift in redox balance. The banded iron formations taper off. Red beds appear, indicating oxidized soils. Traces of atmospheric oxygen, minute but detectable, are preserved in minerals locked into stone. There's no single moment, no boundary line, only the slow, uneven crescendo of a gas changing everything. It is not dramatic, yet it is irreversible. The Earth before oxygen is not the same as the Earth after. Modern analogues help us imagine these ancient transitions. In stratified lakes, microbial mats today show similar patterns: oxygenated upper layers, anoxic depths, metabolic zoning by light and chemistry. In sulfidic springs, bacteria form complex communities where phototrophs, sulfur reducers and fermenters live side by side, each occupying a chemical niche. These systems are not fossils but echoes. They preserve the logic of ancient ecosystems, not their exact species but their strategies. And still some questions resist clarity. Why did it take so long for oxygen to accumulate? Why did cyanobacteria evolve when they did and not earlier? What controls the pace of atmospheric transformation? Was the rise of oxygen continuous or punctuated, steady as a tide, or spiked like a storm? The answers remain elusive. The geological record is incomplete. Biological signals are entangled with abiotic noise. Some researchers propose feedbacks involving phosphorus cycling tectonics, volcanic activity. Others point to gene regulation, the spread of key enzymes, or even global productivity thresholds. The cause may not be singular. It may be a confluence of biological persistence and geological readiness cells, emitting oxygen for millions of years before the planet was finally prepared to keep it. What is certain is that life did not plan for oxygen. It emerged as a byproduct, an unintended consequence of photon capture and water splitting. And yet this byproduct reshaped the world. Oxygen allowed new forms of energy harvesting, new ways of building and maintaining cells. It paved the way for eukaryotes, for multicellularity, for organisms that could grow larger, move faster, think longer. The transformation was neither moral nor inevitable. It was metabolic. And metabolism, like erosion, is both relentless and indifferent. Cyanobacteria remarkably continue unchanged in many ways. They still live in shallow waters, still build stromatolites, still fix carbon and emit oxygen. They have endured extinction events, climatic upheavals, global shifts. Their genomes show both deep conservation and inventive adaptation. Some have partnered with fungi to form lichens. Others live inside plants as endosymbionts. One long ago entered into a eukaryotic cell and became the chloroplast, a symbiotic legacy of the great oxygenation. But most remain free-living, unassuming, reproducing by binary fission in puddles, lakes, oceans, and soil crusts. Their continued presence underscores a broader truth. Evolutionary success is not about novelty, but fitness to environment. The same metabolism that once poisoned the planet now sustains it. The same pigments that absorbed ancient sunlight still catch photons today. That this lineage could reshape the atmosphere and yet remain largely unchanged is a reminder that influence is not always accompanied by transformation. Some revolutions arrive quietly exhaled over eons by bacteria with no voice, no vision, only a persistent need to grow. Even as oxygen establishes itself in the atmosphere, even as the old anaerobic worlds fracture, and new metabolisms emerge, the bacterial domain does not resolve into clarity. Instead, it continues to branch silently without narrative into a sprawling array of lineages. Some drift towards specialized functions, others diversify broadly. New phyla arise not with announcements, but with subtle molecular difference. Proteobacteria, actinobacteria, firmicutes, bacteroidetes, chlorobi, spirochaetes, each unfolding according to its own genomic logic, its own environmental circumstance. There are no explosions of diversity in the dramatic sense, only the slow arithmetic of adaptation. A slight change in membrane composition, a novel transporter protein, a duplication of a regulatory gene, these are the levers by which new lineages emerge. And yet across geological time, these small modifications accumulate, transforming microbial populations into vast, functionally distinct domains of life. Today, these phyla define bacterial diversity, though the labels are human constructs layered over much older realities. The distinctions are molecular, not visible. To the eye, most bacteria remain nearly indistinguishable rods, coxy spirals, filaments. It is only in the genome, in the chemistry of metabolism and the architecture of replication, that differences reveal themselves. The irony is that despite this enormous divergence, much of the bacterial cell remains conserved. Ribosomes, tRNA synthetases, core enzymes, DNA replication machinery. These components vary only modestly between phyla. It is the metabolic appendages, the regulatory networks, the ecological interfaces that shift most. In this, bacteria exhibit a fundamental pattern: conserve what works, diversify what interacts. A kind of evolutionary centripetal force draws the essentials inward, while innovation blooms outward at the periphery. This architecture is not planned. It is the consequence of constraint and opportunity mutation tempered by selective pressure. Over time, lineages develop unique signatures. Some evolve thick peptidoglycan cell walls, becoming gram-positive. Others favor thinner walls with an outer membrane, adopting a gram-negative configuration. These distinctions, later formalized in laboratory staining techniques, reflect deep evolutionary forks. And yet even these categories blur when horizontal gene transfer is considered. A gram-negative bacterium might carry gram-positive genes. A filamentous form might evolve in multiple lineages independently. Morphology, once assumed to be taxonomically significant, reveals itself to be evolutionarily plastic. Phylogenetic reconstruction becomes both a science and a riddle. With the advent of molecular sequencing, especially of the 16S RNA gene, scientists begin to map relationships not through morphology, but through conserved molecular scaffolding. The 16S gene becomes a molecular fossil, changing slowly, yet revealing lineages that diverge billions of years ago. The resulting trees are strange and branching, sometimes elegant, sometimes ambiguous. The deeper one looks, the less resolution appears. At shallow depths within a genus or species, relationships are clear. But at deeper nodes, the branches widen, loop, vanish. The base of the bacterial tree remains unresolved, uncertain. Multiple phyla seem to emerge in parallel or from a common ancestor whose identity dissolves under scrutiny. The root of the tree, that singular imagined origin point, remains speculative. Some suggest it lies among thermophilic lineages, perhaps near the Aquificae or Thermatogae. Others argue for a photosynthetic origin, a cyanobacterial root. Still others posit that the root is unknowable, obscured by the very processes lateral transfer, gene loss, convergence that define bacterial evolution. There is quiet tension in this uncertainty. We classify bacteria as though the boundaries are clear, but nature resists our schemas. Lineages do not always cohere neatly. Families and orders may be polyphyletic. A genus may encompass vast metabolic diversity, while closely related species may inhabit entirely different ecosystems. What appears unified by name may be divergent by function. Taxonomy becomes both necessary and artificial, a scaffolding to hold our understanding but not a mirror of nature's own categories. Metagenomics deepens and complicates this picture. As researchers begin sequencing environmental DNA directly bypassing the need to culture organisms in the lab, they discover a staggering range of bacterial lineages that do not fit existing classifications. Entire phyla are proposed based on genomic fragments, CPR groups, candidate divisions, uncharacterized branches that expand the tree into uncharted territory. Some of these lineages possess minimal genomes lacking many of the canonical genes considered essential. Others harbour novel metabolic pathways, unknown transporters, unusual replication mechanisms. They defy assumptions not by being exotic but by being invisible, present all along in environments never probed, unable or unwilling to grow under laboratory conditions. This raises an epistemological paradox. The more we sequence, the more we realize we do not know. The tree of life, once imagined as a tidy diagram of known species, becomes instead a sprawling, incomplete network. Each new environmental sample adds more tips than it resolves branches. Diversity accelerates. Resolution lags. It becomes clear that what we have studied, what we call model organisms, represents only a sliver of total bacterial diversity. Most of life even today remains uncultured, unclassified, and largely uninterpreted. The microbial majority lives beyond the reach of our tools, yet it forms the metabolic scaffolding of Earth's biosphere. Even within well-studied phyla, surprises persist. Proteobacteria, perhaps the most diverse bacterial phylum, includes phototrophs, nitrogen fixers, pathogens, symbionts, sulfur reducers and methane oxidizers. It spans aquatic and terrestrial habitats from hydrothermal vents to human guts. Actinobacteria, best known for their complex secondary metabolites and soil-dwelling habits, also include marine species pathogens and extremophiles. Bacteroidetes, once considered gut specialists, turn up in ocean sediments and thermal springs. The story is one of versatility, not confinement. Phylum does not equal function. Ecology does not align neatly with ancestry, and still classification proceeds. Not because we believe it is perfect, but because it is useful. It allows comparison inference communication. It lets us say, however tentatively, that this bacterium resembles that one, that these genes co-occur, that these metabolic capabilities cluster. It is a language of approximation, not truth. And in a domain as vast and shifting as bacteria, approximation may be the best we can hope for. A soft irony lingers in this work that we expend such energy naming what does not name itself. That we build trees for organisms that drift horizontally, that we draw boundaries around genomes that reshape with every generation. And yet in this task there is a kind of reverence. To study bacterial lineages is to listen for the planet's most ancient murmurings. To classify is to trace the outlines of life's oldest experiment, an experiment still unfolding. Far from the well-lit waters where cyanobacteria shimmer at the ragged margins of Earth's crust, where light never reaches, another realm of bacteria quietly thrives. These are the thermophils' lineages, adapted not to calm lakes or sunlit shallows, but to scalding heat, to mineral-laden springs, to the deep fissures of hydrothermal vents, where magma warms seawater into near boil. In these habitats, temperature exceeds what was once thought tolerable for life, yet bacteria persist not as exceptions, but as ancient specialists built to endure where proteins should denature and membranes should dissolve. They cluster on black smoker chimneys, where superheated fluids rush into cold ocean depths. They grow in acidic runoff from volcanic slopes in geysers tinted yellow and red by sulfur and iron. Some exist in boiling mud, others beneath kilometers of rock thriving in aquifers heated by geothermal gradients. They do not flinch at 70 dig or even 90. They are not only survivors of heat, they are products of it shaped by the very stress that would unmake most biological systems. The core of thermophilic adaptation lies not in resistance, but in refinement. Proteins are stabilized by increased ionic bonds, tighter folding hydrophobic cores. DNA is wrapped and protected by specialized binding proteins shielded from thermal damage. Membranes incorporate saturated lipids, reducing fluidity and leakage. Enzymes, too, evolve specificity for high-temperature function. They lose flexibility at room temperature, but in their native heat they are swift and precise. These adaptations are not accidents. They are the quiet products of selection operating over millions of years in one of the planet's oldest environments. And yet, despite their complexity, they remain distinctly bacterial. No nucleus, no organelles, just robust machinery humming at temperatures where most life falls apart. Some of these thermophils form entire ecosystems with other extremophils, archaea, protozoa, and viruses within chimney structures or deep crustal fractures. Here, chemosynthesis replaces photosynthesis. Energy comes not from light, but from the oxidation of hydrogen, sulfur, methane, or iron. Carbon dioxide remains the carbon source fixed not by Rubisco, but by alternative pathways, the reductive acetyl-CoA pathway, the reverse TCA cycle. In these systems, bacteria do not consume organic matter. They create it, building biomass from raw chemical gradients in darkness. They form mats, filaments, and biofilms along vent walls, establishing intricate trophic chains where light has never been. Their growth is slow, yet persistent. Some replicate only once every few days. Others enter dormancy waiting for temperature or chemistry to shift. Time flows differently here. A single community may endure for centuries, adapting not through speed but through depth. The evolutionary placement of thermophiles has long intrigued researchers. Some propose that the last universal common ancestor, Leuca, was itself a thermophile born near hot vents, and later giving rise to mesophilic descendants. Others argue that thermophily evolved multiple times with heat-adapted lineages diverging from moderate temperature ancestors. Genomic analyses offer mixed signals. Some core genes in thermophilic bacteria resemble those of archaea, more than other bacteria, suggesting deep divergence or lateral transfer. Heat-stable enzymes, once thought to be ancestral, may have emerged convergently. The tree of life becomes clouded at its roots, with thermophilic branches rising from multiple directions. This ambiguity reveals a broader truth. Extreme environments are not evolutionary backwaters, but frontiers. Thermophiles are not fossils of a hotter past. They are experiments in a different present. Their genomes show both conservatism and creativity. Many carry small, efficient chromosomes with dense coding regions. Others harbour plasmids, transposons, or integrated phage sequences. Lateral gene transfer is common even here. Genes for metal resistance, heat shock proteins, or alternative energy metabolism pass between species aided by viruses or conjugative elements. The boundaries between lineages blur, as they often do in bacterial evolution. Identity is fluid, function paramount, and yet thermophils remain largely invisible in daily human experience. We do not see them on skin or soil. We do not taste them in food or water. But their impact is quietly immense. Their enzymes, particularly DNA polymerase, have revolutionized biotechnology. The polymerase chain reaction PCR, a foundation of molecular biology, depends on a heat-stable enzyme derived from Thermus aquaticus. Other thermophilic proteins enable industrial processes from biofuel production to food processing. We use their molecular adaptations not as novelty but as tools. Their extremity becomes our utility. Still, questions remain. How deep into the crust does life go? What are the limits of temperature, pressure, or pH for bacterial metabolism? Deep biosphere studies continue to uncover bacteria in boreholes, gold mines, and geothermal wells. Some live kilometers beneath the surface where pressure is immense and nutrients scarce. They survive on hydrogen generated by water-rock interactions, or on dissolved organic compounds that have percolated over millennia. Their populations are small, their growth minimal, but they endure. Some may have generation times measured in centuries. Others may be relic cells still viable after tens of thousands of years preserved by stasis rather than reproduction. These revelations reshape our understanding of planetary habitability. Life, it seems, requires less than once assumed. Not warmth, not light, not even abundant carbon. Just energy gradients, water, and molecules capable of self-replication. Thermophilic bacteria embody this minimalism. They show that life is not fragile but adaptable, able to thrive not in spite of harsh conditions but because of them. The Earth's interior, once imagined sterile, is now viewed as a vast microbial reservoir, perhaps rivaling the surface in biomass. Most of it remains unexplored, its diversity unknown. There is a poetic symmetry in this subterranean biosphere. While the surface evolves toward complexity, multicellularity, nervous systems, consciousness, the deep crust harbours simplicity, persistence, and chemical elegance. Bacteria that may not have changed for billions of years still metabolize beneath our feet. Their world is one of slow motion of gradients rather than events. No extinction has reached them. No sunlight guides them. They exist on the edge of detectability, humming with ancient potential. It is tempting to see these thermophils as remnants, as survivors of an early Earth no longer present. But that would be a mistake. They are not relics. They are modern lineages, fully contemporary, fully adapted. Their ancestors may have once pioneered life in a hotter world, but the thermophils of today are not merely echoes. They are active participants in the biosphere, shaping geochemistry, cycling elements, influencing planetary energy budgets. Their silence is not absence. It is stability. So we return to their chimneys, their springs, their fractures. We watch mineral crusts form where their colonies gather. We measure heat and pH, trace isotopic signatures, decode their genes, and we learn slowly that life does not require moderation. It only requires possibility. Bacteria in their thermophilic forms remind us that the boundaries of biology are not fixed. They are experimental, expansive, quietly unfolding in places we once thought empty. While some bacterial lineages sprawl into genomic complexity, accumulating regulatory sequences, accessory genes, and layers of adaptive nuance, others move in the opposite direction toward radical minimalism. In hidden niches where stability reigns and nutrients flow predictably, certain bacteria begin to shed what they do not need. Genes disappear, metabolic pathways contract, redundancies vanish. What remains is a stripped-down genome reduced to its core functions: replication, transcription, translation, a few specialized transporters, and the bare minimum required for survival. These minimalists do not seek simplicity. It arrives as an evolutionary drift, a response to obligate symbiosis or environmental constancy. In the intestines of insects, within the cells of amoebae, or in the cytoplasm of plant tissues, some bacteria become dependent not just ecologically, but genetically on their hosts. They no longer fix nitrogen or synthesize amino acids. Instead, they absorb what the host provides, outsourcing functions they once performed independently. The result is an astonishing economy: genomes with only a few hundred genes, far smaller than those of free-living relatives. Carcinella buchnera wolbachia, each a quiet example of bacterial downsizing a cellular whisper compared to the orchestral complexity of others. The irony is not subtle. In a domain often celebrated for its diversity and adaptability, the smallest genomes are found not in the most primitive bacteria, but in those that have evolved into specialization. Reduction is not regression. It is refinement evolution by subtraction rather than addition. These bacteria are not failing. They are succeeding precisely because they have given up independence. Their hosts offer a buffered environment, removing the need for stress responses, environmental sensing, or broad metabolic versatility. What is lost is no longer necessary. In evolutionary terms, that which is unnecessary becomes a liability. Genes require replication, transcription, regulation. Maintaining them costs energy. If their products are no longer used, selection favors their removal. Over generations, deletions accumulate. Pathways disappear. What remains is lean, focused, efficient. Such reduction often leads to obligate symbiosis. The bacterium can no longer survive outside its host. Its genome lacks key functions for environmental survival. In turn, the host may come to rely on the bacterium for certain nutrients or cofactors. This mutual dependency can become so tight that the boundary between organism and organelle blurs. Some symbionts begin to resemble mitochondria or chloroplasts' endosymbiotic structures with bacterial ancestry. The line between partner and part dissolves, and yet these reduced bacteria are still bacteria. They divide, mutate, respond to selection. Their minimal genomes are not inert. They continue to evolve, though more slowly constrained by smaller populations and less genetic redundancy. Studying these lineages is challenging. Many cannot be cultured outside their host. Sequencing their genomes often requires metagenomic techniques or single-cell methods. Annotation is difficult, so many genes are missing that standard pipelines fail. Yet what we do uncover is revealing. Minimal genomes show us what is essential for life, at least under specific conditions. They provide a baseline, a lower bound on cellular autonomy. They challenge our assumptions about complexity and necessity. A bacterium with only 160 genes can still grow, divide, pass on its lineage. It does not move or hunt or sense light, but it survives generation after generation in a narrow, stable niche. At the other end of the spectrum are the maximalist bacteria, with large genomes, often over 10 million base pairs encoding vast metabolic potential. Streptomyces, Myxococcus, Pseudomonas organisms that thrive in changing environments, compete with others, produce secondary metabolites, form complex colonies. Their genomes are sprawling toolkits rich in regulatory elements, signal transduction systems, biosynthetic gene clusters. They're prepared for change, for competition, for surprise. Between these two extremes, minimalists and maximalists, lies the broad middle where most bacterial genomes cluster. But the contrast reveals something fundamental, that genome size in bacteria is not a measure of advancement or success. It is a reflection of strategy. Simplicity is not failure, it is adaptation. And adaptation in bacteria is not always gradual. Gene loss can occur rapidly under selective pressure. Reductive evolution may begin within a few thousand generations if conditions remain stable. Pseudogenes accumulate, then disappear. Mutations in DNA repair pathways accelerate drift. In symbiotic lineages, bottlenecks and small population sizes enhance genetic drift, allowing mildly deleterious mutations to persist. The result is not just gene loss, but a change in evolutionary tempo. Selection weakens, constraint loosens. The genome slides toward a new equilibrium, not optimized in the classical sense, but stable, persistent, adequate. There are still mysteries here. How far can reduction go? What is the absolute minimal set of genes required for cellular life? Synthetic biology has attempted to answer this building minimal cells with curated genomes. The results vary, but a common number hovers around 300 essential genes for basic function in a controlled environment. Natural minimalists often fall below this threshold, relying on their host to fill in the missing functions. In doing so, they redefine what autonomy means. A cell may no longer be a unit of self-sufficiency, but of integrated dependence. This dependence is not always symmetrical. Some hosts rely deeply on their bacterial partners, others do not. In many cases, the symbiont is maintained only because it has not yet been replaced. Evolution, after all, is not always efficient. It is incremental. A symbiont may persist simply because no better solution has emerged, or because replacement would require intermediate steps too costly to survive. As such, many minimal bacteria are evolutionary holdouts, intermediate stages on a spectrum between independence and integration. Beyond animal and plant symbionts, minimal genomes also appear in extreme environments. Some bacteria in deep aquifers, nutrient-depleted sediments, or hypersaline lakes exhibit genome reduction, not due to symbiosis but to energy limitation. In these habitats, every unnecessary gene becomes a cost. Cells divide, rarely conserve energy-limit biosynthetic pathways. Their minimalism is not social but environmental, a solitary austerity rather than a partnership. Modern sequencing continues to reveal new minimalists, many hidden in obscure ecosystems. Some are predicted only from environmental DNA with no cultured representatives. Others are inferred from fragmentary scaffolds, yet show consistent patterns, small genomes, few regulators, limited flexibility. These lineages challenge the completeness of our bacterial tree. Are they old, recent derived, or ancestral? The phylogenies are often shallow, distorted by high mutation rates and gene loss. Their placement remains tentative, but their existence is certain, and their persistence undeniable. In the end, the minimalist bacterium is not an outlier. It is a reminder that survival does not always require abundance, that life can thrive not by doing more, but by doing less, precisely quietly, and with just enough to endure. Across the deepening layers of microbial time, even as some lineages strip themselves down to minimal function, others proliferate not by size or number, but through their biochemical diversity. The bacterial domain, ever adaptive, begins to explore the full range of Earth's elemental palette. Sulfur, nitrogen, hydrogen, carbon monoxide, manganese, arsenic, methane. Each becomes, in different contexts, a substrate, an electron donor, a terminal acceptor. There is no singular way to live, no canonical metabolism. Instead, there is a forest of metabolisms, dense and branching, filled with lineages that harvest energy in ways that seem improbable to multicellular life. Some bacteria oxidize hydrogen sulfide into sulfate. Others reduce sulfate back into sulfide. One fixes nitrogen from the atmosphere. Another oxidizes ammonia into nitrate. Some grow phototrophically in the light of thermal pools. Others ferment organic compounds in lightless bogs. The combinations are vast, the biochemistry intricate, and yet each pathway is rooted in the same basic logic: electrons must move, and in that movement energy can be captured and used to power life. This metabolic diversity is not decoration, it is survival. In a world where conditions shift across gradients of light, temperature, pressure, salinity and chemical composition, the ability to exploit a wide range of energy sources becomes an evolutionary advantage. Metabolic plasticity allows bacteria to occupy virtually every environment on the planet, from oxygen-rich river mouths to anoxic mud from acid mine drainage to the alkaline waters of soda lakes. Even within a single habitat, bacteria differentiate spatially by metabolism. In a sediment column only a few centimeters deep, one can find oxygen respirers near the top nitrate reducers, just below, followed by manganese reducers, iron reducers, sulfate reducers, and methanogens near the bottom. Each layer is defined not by species but by redox potential. Life arranges itself not along a hierarchy of form but a gradient of chemistry. And yet, for all their functional ingenuity, many of these bacteria remain unnamed, uncultured, unclassified. They are known by what they do, not by what they look like. Metagenomic surveys of soils, sediments, oceans and aquifers reveal immense functional gene diversity. Gene clusters for sulfur oxidation, nitrogen fixation, carbon monoxide dehydrogenation, found in unexpected lineages in surprising places. This genomic data paints a portrait of bacterial life as less a series of trees and more a biochemical mosaic. Traits jump between lineages. Pathways reappear in distantly related clades. Convergent evolution, gene duplication, horizontal transfer all reshape the metabolic landscape. The result is not disorder, but a dynamic equilibrium. Ecosystems stabilize through functional redundancy. If one lineage disappears, another with a similar pathway may take its place. It is not the organism that matters most, it is the function it performs. This observation has transformed microbial ecology. Scientists now speak of functional guilds, groups of organisms defined not by ancestry, but by shared metabolic roles. Iron reducers, methanotrophs, nitrifiers, denitrifiers. These guilds persist even as their membership changes, and because of this, microbial communities are resilient. Disturb a system, and the species composition may shift, but the chemical cycling continues. A sulfur oxidizer is replaced by another. A methanogen finds new partnerships. The forest of metabolisms bends but does not break. One of the most studied examples of such guild dynamics is the nitrogen cycle. In soils and oceans, nitrogen undergoes a complex series of transformations: fixation of atmospheric N2 into ammonia, nitrification into nitrite and nitrate assimilation into biomass, and eventual denitrification back to N2 gas. Each step is performed by different bacteria. Some obligate, some facultative, some free-living, others symbiotic. The genes involved, nifamo narnir nor nos, form a distributed system scattered across taxa, yet tightly coupled in ecosystem function. When conditions change, oxygen drops, carbon becomes limiting, different steps become bottlenecks. Communities adjust, upregulate, downregulate, reconfigure. The cycle continues shaped not by hierarchy but by feedback. Another elegant example lies in methane cycling. In wetlands, sediments, and deep marine clathrates, methane is both produced and consumed by bacteria. Methanogens, archaea that produce methane under anoxic conditions, are complemented by anaerobic methane-oxidizing bacteria ANME, which consume methane in partnership with sulfate reducers. In oxygenated waters, aerobic methanotrophs use methane as a carbon and energy source. These systems are tightly balanced. Too much methane and greenhouse effects intensify. Too little and microbial communities lose a vital energy source. The stability of the cycle is not due to oversight, but to the distributed logic of bacterial metabolism. Regulation arises from interaction, not command. Still, there are processes we do not fully understand. Arsenic-based metabolisms, for example, persist in extreme environments, hypersaline lakes, geothermal springs, but their enzymes and regulation remain elusive. Manganese reduction, widespread and geochemically significant, is poorly characterized at the genetic level. Even well-known pathways like sulfate reduction vary dramatically between lineages with different enzymes, cofactors, and regulation. Our models are incomplete. Our assumptions are often based on a handful of cultivable representatives, while most of the metabolic forest remains unexplored. It is tempting to see this biochemical diversity as overwhelming, but it is in fact elegantly constrained. Bacteria do not invent new chemistries from nothing. They repurpose, refine, recombine. A protein that once transferred electrons from hydrogen to NAD+, becomes adapted to handle iron, sulfur, or arsenic. Enzyme families diversify. Gene clusters evolve modularity. Regulation is layered onto existing frameworks. There are only so many thermodynamically viable reactions on Earth. What varies is how bacteria access them, how they couple them to growth, how they integrate them into community function. In modern biotechnology, this flexibility is harnessed. Bacteria are engineered to degrade pollutants, fix nitrogen in crops, produce fuels. Synthetic biology draws on the microbial gene pool to construct new pathways. But even here, the natural precedents often outstrip our imagination. What seems novel to us has usually been done somewhere by some lineage at some point in Earth's long microbial history. The forest of metabolisms is not new. It is ancient, rooted in the earliest innovations of life expanding ever since. There is a final irony in this diversity. Despite its complexity, it remains largely invisible. The vast majority of metabolic processes on Earth occur at scales too small to see, too slow to notice, too common to provoke awe. Bacteria fix nitrogen in roots, cycle sulfur in sediments, oxidize iron in groundwater, all without spectacle. Their chemistry is constant, quiet, and essential. Without them, ecosystems collapse. Biogeochemical cycles halt life as we know it ceases. And yet they remain uncelebrated, faceless, structureless, working beneath awareness. To walk through a forest, drink from a stream, breathe air rich in oxygen, is to move through bacterial legacy. To study metabolism is to trace the contours of ancient ingenuity, life crafting itself not from narrative but from necessity. And in every gradient, every molecule passed between cells, the bacterial world continues not with grandeur but with persistence. In the quiet pursuit of order beneath the microscope and within the genome, scientists attempt to arrange bacterial diversity into trees, into clades, into an ancestral flow. But the deeper they search, the less clear the lines become. Nowhere is this more apparent than in the question of bacterial origins of the deepest nodes on the bacterial phylogenetic tree. Unlike the relatively clear divergence between domains bacteria, archaea, and eukarya, the internal structure of the bacterial domain remains blurred, tangled, uncertain. Every attempt to root the bacterial tree stumbles on the same paradox. The more ancient the divergence, the more obscured it becomes by the very processes that define bacterial life. Horizontal gene transfer, differential gene, loss variable, evolutionary rates, and the mosaic nature of genomes erode the clarity of lineage. There is no single undisputed point from which all bacterial diversity springs, no definitive root, no fossilized ancestor, no preserved code untouched by recombination. Instead, there is a forest of deep lineages, each with its own complexity, each appearing almost simultaneously in the geological and genomic record. Some of these early branches are thermophilic, thriving in high-temperature environments, suggestive of an origin near Earth's geothermal features. Others show evidence of photosynthesis, carbon fixation, or complex redox metabolisms implying not a single ancestral metabolism, but a suite of strategies already in play. Genomic reconstructions offer competing hypotheses. One tree places the root near the firmicutes, another places it among the chloroflexi. A third suggests the deepest branch lies near the candidate phyla radiation, a group of ultra-small genome-reduced bacteria discovered only through metagenomics. Each model depends on assumptions on gene selection, on models of sequence evolution, on interpretations of early metabolic traits. None are definitive, each is provisional. This ambiguity reflects more than just technical limitation. It may point to a deeper truth that the origin of bacterial lineages was not a clean branching event, but a prolonged period of gene sharing among early protocells. In this view, the earliest forms of life did not inherit their genes vertically in a linear fashion, but acquired them laterally, building functionality piece by piece from a communal gene pool. The first cells may have existed less as species and more as networks, fluid assemblies of membranes and reactions exchanging components freely. The transition to modern bacteria with relatively stable genomes and membrane-bound integrity may have been gradual, marked by increasing genetic insulation, the rise of error-correcting mechanisms, and the compartmentalization of function. If so, then searching for a root may be missing the point. The tree in its earliest moments was not yet a tree. It was a web, a field, a swarm of possibilities. Modern genomic studies of deep branching lineages reveal further complications. The candidate filler radiation CPR, a recently characterized group comprising dozens of phyla, includes bacteria with tiny genomes, reduced metabolic capacities, and unusual cellular structures. Many are predicted to be symbionts or episymbionts, dependent on host organisms for essential functions. Their placement in the bacterial tree is uncertain. Some trees place CPR as a basal group, others suggest they are highly derived. Their gene content is strange, lacking many universal genes but containing unusual adaptations. They do not fit our models, and yet they may hold clues to early bacterial evolution, particularly the shift from communal networks to individualized cells. Other deep lineages, such as the Thermatogoles aquifici and Synergistetes, further complicate the picture. Their thermophilic lifestyles, ancient-looking metabolic pathways and gene content suggest early divergence, yet their genomes also show evidence of extensive lateral gene acquisition, including from archaea. Are they primitive, or are they the result of convergence and gene swapping in extreme environments? The evidence is ambiguous. Their ribosomal genes tell one story, their metabolic genes another. Each lineage appears as a palimpsest old signals, overwritten by new adaptations. This confusion extends even to our definitions. What is a bacterial phylum? At what point does sequence divergence or metabolic distinctness justify a new category? Taxonomy, though anchored in molecular data, remains human-made. The number of recognized bacterial phyla has grown steadily, especially with the expansion of metagenomics. Some estimates place the number at over 100, with new lineages proposed regularly. Many are known only from environmental samples, never cultured, never observed, inferred only from their DNA. They may be common in groundwater soil or sediments, yet invisible to classical microbiology. Their names, candidatus groups, provisional acronyms hint at their provisional status. They are real, but undefined. This explosion of diversity raises a deeper question: how much of the bacterial domain remains undiscovered? The answer seems to be most of it. Surveys of environmental DNA routinely uncover novel lineages, many with low sequence identity to any known organism. Each new ecosystem sampled deep ocean crust, permafrost, high-altitude clouds reveals unfamiliar branches. The bacterial tree is not nearing completion. It is still in its infancy, and beneath each branch new branches wait. The structure is not static. It is fractal, unfolding. Even within well-known groups, surprises arise. New species, new genes, new functions. A bacterium once thought to be obligately anaerobic is found to tolerate oxygen. Another long assumed to fix nitrogen does not. Our classifications trail behind the reality they aim to describe. We label by what we see, but what we see is partial, filtered through methods and expectations. As such, the bacterial tree is not a map but a hypothesis, a model to be revised, challenged, undone. And still we persist in drawing it, not because we believe it to be final, but because it is a way to organize the overwhelming complexity, to place a gene in context, to compare organisms, to ask questions. Even a flawed tree can reveal patterns, the clustering of phototrophs, the distribution of sulfur metabolizers, the independent emergence of multicellularity in filamentous bacteria. Evolution leaves shadows. The tree is how we trace them. One quiet irony remains. In seeking the origin of bacterial diversity, we must often turn away from cultivated strains, those grown, isolated and studied for decades, and look instead to the uncultured, the obscure, the lineages known only by code. The bacteria that fill textbooks are not the deepest or most representative. They are the most convenient. The real story of bacterial origins lies elsewhere, in dark sediment, in deep aquifers, in ancient rocks in the unknown, and so we continue to search, to sequence, to arrange, not for closure but for a clearer ambiguity, a more refined uncertainty. The tree may never resolve at its base, but the act of seeking it reveals the elegance of microbial history, a world without roots yet teeming with life. If the tree remains unresolved at its root, then perhaps it is not the origin of bacteria that best defines them, but the way we learn to compare them. In the absence of clear ancestry or fossilized morphology, classification in bacteriology has become increasingly reliant on molecular comparison, an approach that does not presume shared form, only shared sequence. The genetic code becomes both mirror and map. Instead of shapes or behaviors, we trace patterns in nucleotides, the order of bases in ribosomal RNA, the presence or absence of specific operons, the arrangement of conserved housekeeping genes. These comparisons form the scaffolding of modern bacterial taxonomy, transforming invisible differences into measurable relationships. But this too is not straightforward. Genes evolve at different rates. Some mutate slowly, retaining deep phylogenetic signals. Others are fast-changing, reshaped by environmental pressures, horizontal transfer or gene loss. The result is a patchwork of signal and noise, a challenge to every attempt at consistent classification. The most foundational tool in this effort is the small subunit ribosomal RNA gene 16S, rRNA in bacteria. It is ancient, present in all known bacterial lineages, and contains both conserved and variable regions, allowing comparison across a wide evolutionary scale. For decades, 16S sequencing served as the gold standard of bacterial identification. Species were grouped by sequence similarity thresholds, defined trees drawn. But over time, limitations became clear. The gene is sometimes duplicated within genomes. It may be too conserved to resolve closely related taxa, or too variable to compare distant ones. And most crucially, it represents only one thread in the vast fabric of the genome. A single gene, no matter how universal, cannot capture the full complexity of an organism's biology. To address this, modern approaches incorporate multi-locus sequence analysis, MLSA, genome-wide average nucleotide identity ANA, and whole genome phylogeny. These methods compare not just one gene, but many or even all, aligning hundreds of conserved proteins, calculating divergence across entire chromosomes. ANI, for example, measures the mean similarity of all orthologous genes between two genomes. Values above 95% often indicate members of the same species. Lower values suggest distinct taxa. This numerical approach, grounded in comparative genomics, offers a more robust framework for classification. It is not perfect, but it reflects evolutionary relatedness more faithfully than morphology ever could. Yet even these refined tools confront bacterial peculiarities. Genome size varies widely. Some bacteria carry vast accessory gene pools, plasmids, prophages, genomic islands that differ even between strains of the same species. Others possess streamlined chromosomes with little redundancy. Two bacteria may share high ani in their core genomes, yet differ dramatically in lifestyle and function due to variable regions. The question arises, what defines a species? Is it genomic similarity, ecological role, metabolic capacity, or reproductive isolation, if such a term even applies to asexual organisms? In bacteria, there is no consensus. Definitions remain operational, pragmatic, adapted to context. Some researchers define species based on ANE thresholds. Others use digital DNA-DNA hybridization, phylogenomic placement, or ecological coherence. There is no single answer, and perhaps there does not need to be. This ambiguity reflects not failure but flexibility. Classification is a tool for understanding, not a law of nature. It serves to group organisms with shared properties to infer relationships to predict traits. And bacteria, more than any other domain of life, challenge the rigidity of categories. They swap genes across taxonomic boundaries. They evolve in rapid mosaic bursts. They blur the lines between clade and community, between lineage and lifestyle. Still, classification continues not to impose order, but to trace patterns. Some patterns are clear. The phylum Proteobacteria, for instance, contains an astonishing diversity of metabolic types: sulfur oxidizers, nitrogen fixers, aerobic heterotrophs, phototrophs, and pathogens. Yet within this vast clade, gene content and structure follow predictable trends. Certain families cluster together by genome architecture, GC content regulatory motifs. The same holds true for other major phyla Actinobacteria, with their high GC content and complex secondary metabolites. Firmicutes with their thick cell walls and low GC genomes. Bacteroidetes, often associated with animal guts and polysaccharide degradation. These associations, though broad, provide structure. They allow generalizations to emerge without denying exception. At finer scales, classification enables ecological insight. Within a soil sample, for instance, sequencing may reveal dozens of bacterial taxa. Grouping them into operational taxonomic units, OTUs or amplicon sequence variants, ASVs, allows researchers to track diversity, richness, and evenness across samples and over time. While these units may not correspond precisely to named species, they serve as functional proxies revealing how communities assemble, shift, and respond. In clinical contexts, classification identifies pathogens, tracks outbreaks, monitors antibiotic resistance. In biogeochemistry, it maps the distribution of metabolic guilds. In evolution, it reconstructs the history of gene families. And yet the act of comparison is not only practical, it is also philosophical. To compare bacteria is to ask what makes one organism different from another, what traits matter, what histories count. It is to recognize both unity and divergence, to see in the code not just sequence but story. Each genome is a record of past challenges. Toxins survived environments, entered partners, found enemies, resisted. The presence of a gene is a clue, its absence an echo. To compare is to read between these lines, to find in molecular patterns the residues of ecological and evolutionary choice. Still, classification can be fragile. As new data accumulate, trees shift. Whole clades are renamed, redefined, reclassified. What was once a single genus becomes three. A species is split or merged or relocated. Names change, relationships invert. These revisions may seem disruptive, but they reflect the living nature of science, a willingness to revise, to adapt, to improve. Bacteria do not mind our labels. They do not self-identify as Clostridium or Acinetobacter. They persist indifferent to taxonomic reordering, evolving at scales and in ways our systems only partly capture. In the end, classification is not about perfection. It is about correspondence between genome and function, between lineage and niche, between code and consequence. The better our tools, the more nuanced our groupings, the more precisely we can ask, how does this bacterium live? What does it do? And how does it fit into the fabric of life? That question, however provisional, remains at the heart of microbial systematics. Not to constrain the bacterial world within boundaries, but to illuminate its continuities, its deviations, and its quiet genomic constellations. In the slow breath of geologic time, as bacteria spread across oceans, crusts, and sediments, many disappear without a trace. Their cells degrade, their genes are lost, their lineages vanish, and yet some persist not through continued activity, but through dormancy. Frozen in ice, entombed in salt, embedded in amber or permafrost, certain bacterial cells enter states of suspended metabolism, waiting in silence for conditions to shift. These dormant forms are not artifacts or fossils. They are alive in the most minimal sense intact membranes preserve DNA-halted division. They do not grow, but they endure. In Arctic ice cores, viable bacteria have been recovered after hundreds of thousands of years. In deep-sea sediments, cells buried beneath kilometers of water and silt continue to respire at astonishingly slow rates. One metabolic flicker every few centuries. Inhalite crystals from ancient salt mines' cells persist in brine inclusions, living quietly in a world closed off for geological epochs. Life, it seems, is not only about movement, but about waiting. Bacterial dormancy takes many forms. Some produce spores highly resistant desiccated structures formed when nutrients become scarce. These spores contain a condensed chromosome protective protein's dehydrated cytoplasm and thick layers of peptidoglycan and spore coat. They can survive ultraviolet radiation, extreme heat pressure, desiccation, and chemical assault. Genera like Bacillus and Clostridium are masters of this form, creating spores that remain viable for millions of years under the right conditions. Others enter states of viable but non-culturable VBNC existence. The cells are alive but fail to divide or form colonies under standard conditions. Some reduce their cell volume, synthesize stress proteins, shrink their membranes, and abandon active metabolism almost entirely. In these states, life is held in abeyance like a script memorized but not spoken. The evolutionary logic of dormancy is clear. Environments fluctuate. Nutrient pulses come and go. Extremes of temperature, salinity, or acidity may last for months, years, or millennia. For bacteria whose generation times are short and whose populations are vast, entering dormancy becomes a bet against uncertainty. It spreads risk across time. While some cells continue to divide others, weight held in reserve. If the environment improves, the dormant awaken and repopulate. If it does not, the lineage continues elsewhere or in other forms. This strategy, phenotypic heterogeneity within a clonal population, ensures resilience without requiring foresight. It is not a choice, but a result of molecular regulation, stochastic gene expression, and evolutionary tuning. This dormancy extends beyond survival. It shapes ecosystem function. In soils, vast numbers of bacterial cells are dormant at any given time, forming a microbial seed bank. These latent populations influence nutrient cycling succession and community resilience. When disturbances occur, fire, flood, drought, dormant taxa can re-emerge, restoring lost functions. In marine sediments, subsurface microbial communities persist with extremely low energy inputs. Cell turnover may occur only once every 1,000 years, yet these communities maintain structure, function, and even limited evolution. Their genomes accumulate mutations slowly. Their proteins remain folded. Their cells are not dead, they are patient. Dormancy also complicates our understanding of abundance and diversity. Sequencing surveys often detect DNA from cells that are no longer active. Are they alive? Are they relevant? The distinction is blurred. A dormant bacterium may not contribute to current function, but may re-emerge to shape future dynamics. Its DNA is not merely residue, it is potential. The concept of microbial dark matter includes such dormant lineages, those known from sequence but never cultured, never observed in action. Some may be rare, others may be widespread but inactive. Their role in biogeochemical cycles is subtle, modulated by activation thresholds, environmental cues and community context. One of the great mysteries in this realm is longevity. How long can a bacterial cell persist without dividing? What limits define biological time? Some spores have been claimed to revive after tens of millions of years, though such claims are contested. More conservatively, we know that viable cells have been recovered from ice amber and salt deposits, dating back hundreds of thousands to a few million years. Their survival depends on stability, low radiation, low temperature, low metabolic demand. In these conditions, DNA decay is slow, membranes remain intact, metabolism pauses but damage is minimized. It is not immortality. It is extremely slow degradation. The ability to survive such time spans also raises questions about planetary boundaries. If bacteria can persist in rock ice or salt for geological epochs, then the concept of extinction becomes more complex. Lineages may disappear from active ecosystems but remain entombed elsewhere waiting for thaw or fracture or contact. This is not speculation. Studies of melting permafrost, ancient salt domes and subsurface brines consistently reveal viable cells. Some are closely related to modern taxa. Others appear divergent, ancient, genetically distinct. Their return is not resurrection, it is re-entry. In astrobiology, these findings inspire thought. If life ever existed on Mars or beneath the ice of Europa, it may not be active, but it may be dormant, hidden within rock, surviving radiation and desiccation, awaiting warmth or water. Earth's own microbes have demonstrated that life can slow to a whisper and still remain. A bacterial cell locked in crystal or clay may outlast species, climates, even continents. It is not a romantic endurance, it is molecular pragmatism. Still, dormancy is not stasis. Even within dormant populations, tiny amounts of energy may be expended on repair, maintenance, damage, mitigation. DNA repair enzymes, membrane integrity checks, low-level redox activity all continue at minimal rates. These processes allow survival far beyond the limits of active growth. They are not designed for comfort, only for persistence. In this, bacteria challenge our assumptions about what it means to be alive. Life does not require growth. It does not require consciousness. It requires continuity, a sustained configuration of molecules within a bounded system capable of responding eventually to favorable conditions. There is something quietly humbling about these dormant forms. While animals age, decay, and die, while plants wither and fall, these cells persist. They do not race forward. They wait. In the walls of salt mines, in the cores of glaciers, in sediments laid down when continents were aligned differently, bacteria remain. Some may divide again, some may not. But their presence tells us that life's timeline is not only linear. It loops, pauses, folds back on itself. It waits where it must. In this waiting, bacterial dormancy becomes a kind of memory, not conscious, not directed, but molecular. A record of past environments, past communities, past climates, stored not in data or symbols, but in preserved potential. The cells carry forward a possibility, a message encoded in silence. If conditions permit, the message will resume. If not, it remains inert, but not erased. Beneath our feet, across our skin, within our lungs, mouths and intestines, bacteria persist not as remnants of the distant past, but as quiet inheritors of evolutionary endurance. Every moment they metabolize, divide and interact with the human body, forming complex dynamic ecosystems we're only beginning to understand. The human microbiome, once an abstract concept, has become a central field of study, revealing that bacterial cells in and on our bodies rival or even outnumber human cells. But these bacteria are not invaders. They are inhabitants long embedded in the rhythms of our physiology, co-evolved to inhabit our mucosal linings, our follicles, our gut lumen. Some digest complex carbohydrates we cannot process. Others synthesize vitamins or modulate immune responses. Their presence is not incidental, it is foundational. Yet the bacteria of the modern microbiome carry deep ancestral echoes. Genomic studies reveal that many of our microbial companions belong to ancient lineages shaped by eons of environmental selection before ever encountering vertebrate hosts. Bacteroids, firmicutes, actinobacteria. These genera trace their histories far beyond the appearance of mammals. Their association with animals may be recent compared to their total evolutionary span, but their adaptability is ancient. Some shift freely between host and environment, thriving in soil or water as easily as in intestines. Others have become host-restricted, evolving intimate dependencies, losing genes unnecessary outside the body. Like the symbionts of insects or plants, some human-associated bacteria show signs of reduction, adaptation, specialization. They are not passengers. They are partners, silent, adaptable, ancient. This bacterial presence begins early. Infants acquire microbes during birth from maternal skin, breast milk, the surrounding environment. The gut, once sterile in utero, becomes rapidly colonized. These first microbes help train the immune system, shape intestinal development, and influence metabolism. Over time, the microbiome stabilizes into a characteristic adult profile, though it remains dynamic, responsive to diet, stress, disease, and age. Antibiotics, illness, or changes in environment can reshape it within days, and yet in the absence of disruption, the core community persists. Species fluctuate, but functions are retained. Redundancy ensures stability. Like ancient microbial mats, the human microbiome is layered, zoned, and self-reinforcing. But not all associations are benign. Some bacteria long considered commensals become opportunistic under stress. Others are outright pathogens, invading tissues, evading immunity, causing inflammation, damage, death. And yet even pathogens are not static. Many arise from formerly harmless strains acquiring virulence genes through horizontal transfer. A toxin gene picked up from a phage, a secretion system acquired from a plasmid, a capsule synthesis cluster, transferred from a distant cousin. These modular changes turn symbiont to threat. Evolution does not favor kindness, it favors persistence. And in the host environment, this may mean cooperation, competition, or exploitation. The boundaries are fluid. Pathogenicity is not a trait but a context. In health, bacteria form structured communities, biofilms on teeth, mucosal layers in the gut, low diversity skin populations, adapted to pH, salt, and moisture. These communities communicate, share resources, compete for space. They influence gene expression in host cells, modulate inflammatory responses, even affect behavior and cognition through chemical signaling along the gut-brain axis. While mechanisms remain under study, the idea is increasingly clear: bacterial life does not end at the cell membrane. Its effects radiate outward into tissues, systems, behavior, population health. Yet as always in microbiology, much remains unknown. The diversity of the human microbiome defies easy categorization. Two individuals may share only a small fraction of bacterial species, yet exhibit similar metabolic functions. Genes are more conserved than taxa. Functional redundancy is high. A shift in diet may alter species composition, but keep core pathways intact. This suggests that the microbiome is not a fixed assemblage of names, but a fluid network of roles. What matters is not who is present, but what they are doing. Even rare species present at low abundance may contribute key enzymes or regulatory signals. Modern tools have begun to illuminate this invisible ecology. Metagenomic sequences microbial DNA directly from samples revealing taxonomic profiles and functional genes. Metatranscriptomics captures RNA showing what genes are actively expressed. Metabolomics tracks the chemical products of microbial metabolism. Combined, these approaches offer a view not just of composition but of activity, who is there, what they are doing, and how it changes over time. Yet interpretation is difficult. Correlation is not causation. Many microbes remain uncultured, uncharacterized. Functional predictions are based on annotation databases themselves incomplete, and host variability genetic dietary behavioral adds layers of complexity. Despite this, patterns emerge. Diets rich in fiber support greater microbial diversity. Western diets high in processed food and low in plant polysaccharides tend to reduce diversity and shift populations toward species associated with inflammation. Antibiotic use, especially early in life, can have long-term effects on microbiome composition and health outcomes. Fecal microbiota transplants, once fringe, are now used to treat Clostridioides difficile infections with high success. Probiotics and prebiotics, though inconsistently effective, are under study for a range of conditions from irritable bowel to metabolic disease to mood disorders. The idea is simple: by modulating the microbial community we may modulate the host. But the microbiome is not only a medical frontier, it is also an evolutionary relic, a living trace of bacterial persistence through the vertebrate lineage. Many gut-associated bacteria appear to have co-evolved with their hosts, showing phylogenetic patterns that mirror mammalian evolution. Others have jumped between hosts, opportunistically spreading via contact environment or shared food. The gut, after all, is not a closed system. It is a corridor between the body and the world, lined with cells, mucus, and microbial residents. It is an ecosystem shaped by selection, constraint, and exchange. And in this corridor, bacteria remind us of a deeper truth, that individuality is porous. Our bodies are not discrete entities, but assemblages of human and microbial cells, of host and commensal, of ancient lineages interacting in real time. We are not alone, not even in ourselves. Our thoughts, our health, our biology are shaped by communities invisible yet present in every breath, every bite, every cell layer. To study bacteria is to study the boundaries of self, the continuity of life across form and function. These communities, quiet and unseen, are no less vital than those that formed stromatolites or oxidized iron in early oceans. They are part of the same story, the same ongoing experiment of life adapting to chemical gradients, exploiting niches, persisting through change. And just as bacterial lineages once transformed the planet with oxygen, so now they shape the internal geographies of multicellular hosts, quiet architects of the present. Even now, as the Anthropocene unfolds with its concrete landscapes and climate-altered skies, bacterial lineages continue to evolve, to diversify and to emerge from places still unmeasured, unimagined. While we sequence genomes and label phyla, while we classify, annotate, and model, new branches unfurl in the microbial tree, not as explosions of innovation, but as gentle, steady divergence. A bacterium in a thermal vent quietly acquires a new transporter gene. Another in a peat bog loses a biosynthetic cluster it no longer needs. A lineage long thought uniform turns out under closer scrutiny to be a complex of cryptic species genetically distinct yet morphologically indistinguishable. Evolution does not sleep. Even in the tiniest, most conserved genomes, mutations accumulate. Selection filters, drift reshapes, recombination redraws. And through these slow dynamics, novelty arises not suddenly, but cumulatively layered into the genome like sediment in stone. Much of this diversification remains invisible. It occurs in microbial dark matter, those lineages known only through environmental DNA lacking cultivated representatives or cultured description. These uncultured bacteria dominate many ecosystems deep, soils subsurface, aquifers, marine sediments, oxygen minimum zones, acid mines, saline lagoons. Their DNA appears in metagenomic surveys, their presence inferred by coverage, abundance, co-occurrence. But they remain unnamed, unpictured, uncharacterized. Thousands of proposed candidate phyla have been described, each with its own branch on the tree, its own genomic peculiarities, its own ecological role. Some possess ultra-small genomes, reduced cell structures, and unusual cell envelopes. Others show complex capabilities, novel respiratory pathways, unique adaptations to pressure, salinity, or desiccation. These organisms are not rare. They are simply difficult to reach, to grow, to isolate. They have adapted to conditions that standard laboratory media cannot replicate. Their life strategies are subtle, sometimes syntrophic, dependent on signals or compounds produced by neighboring cells. They are not autonomous in the way we've come to expect, but they are no less real. This continuing diversification raises a quiet irony. While we imagine bacteria as ancient, static, unchanging, their evolutionary pace is often faster than ours. Their populations are vast, their generation times short, their mutation rates steady. They experiment more than we do across space, across time, across chemical possibilities. Their innovations are not always visible as form, but as function, a new enzyme, a different membrane structure, an altered regulatory circuit. And in this experimentation they often outpace our capacity to observe. Many bacterial lineages today are evolving in real time in environments we do not sample, beneath glaciers, in deep biosphere aquifers, in urban stormwater runoff, on plastics drifting through the sea. Their adaptation is not dramatic, it is iterative, a series of molecular adjustments, each nearly invisible, yet in aggregate pushing the lineage into new ecological space. Some of these emerging lineages are being discovered through advances in single-cell genomics. By isolating individual bacterial cells from environmental samples and sequencing their DNA directly, researchers can reconstruct genomes from organisms never grown in culture. These techniques have revealed bacteria with unanticipated complexity, novel CRISPR systems, new types of nitrogenases, unexpected biosynthetic pathways. Others possess minimal systems barely sufficient for replication, suggesting novel symbioses or highly specialized niches. In each case, the lesson is the same: bacterial diversity is not exhausted. It is expanding. Even in well-studied environments, previously unknown lineages continue to emerge. Wastewater treatment plants, for example, host rich dynamic microbial communities essential for nutrient removal and chemical transformation. These systems have revealed dozens of new bacterial groups, some with previously unknown metabolic traits, such as the ability to anaerobically oxidize ammonia or to metabolize complex pharmaceuticals. Urban environments, once considered microbiologically dull, are increasingly recognized as arenas of microbial innovation. Surfaces in subways, hospitals, and homes host bacteria that adapt to cleaning agents, antibiotics, synthetic compounds. These microbes are not inherently pathogenic, but their proximity to human activity and their exposure to selective pressures makes them key players in the evolution of resistance degradation and interaction. Climate change adds further complexity. As temperatures rise, as permafrost thaws, as oceanic oxygen levels decline, microbial communities shift. Some expand, others retreat. Bacteria that were once dormant awaken. Methanogens and methanotrophs respond to shifting carbon flows. Sulfur and nitrogen cycles are altered. Soil respiration increases. Feedback loops are established. These changes are not abstract. They are biochemical, genomic, cellular, playing out in bacterial lineages with long histories and short generation times. Evolution accelerates where stress accumulates. And in this new environmental context, lineages that have persisted in relative stasis may diversify anew. Still, many of these emergent lineages remain uncatalogued. The sheer number of bacterial species is unknown. Estimates range into the millions or billions. Only a fraction have been described. Taxonomic backlogs grow as sequencing outpaces curation. Automated pipelines assign provisional names. Group genomes into clusters suggest tentative affiliations. But each sequence, each genome represents a living cell, somewhere a product of billions of years of molecular inheritance shaped by unique pressures, carrying both ancient and novel traits. Each one contributes to the vast biochemical meshwork that underpins global ecosystems. Their diversification is not a background process. It is the texture of life itself. There is a quiet awe in this realization that as we seek to understand bacterial evolution, we're surrounded by its ongoing expression. It is in the soil beneath our shoes, in the water we drink, in the air we exhale. It is in the biofilms that coat rocks, pipes, leaves, and teeth. It is in the fermentation vats and hydrothermal vents, the coral mucus and glacial melt. It is slow and fast, ancient and contemporary, constrained and creative. It is not a process that happened. It is one that never stopped. To classify bacteria is to capture a moment in motion. To study them is to observe not a relic, but a river. Their lineages twist, merge, fork, vanish. Some persist almost unchanged for billions of years. Others bloom and fade within centuries. Evolution among bacteria is not linear. It is rhythmic, recursive, and responsive, and it continues in silence and in number whether we look or not. In this unfolding there is no final form, no apex, no completion, only the endless, quiet diversification of bacterial life adjusting to gradients, discovering new chemistries, reaching into unoccupied niches. From the earliest anoxic oceans to the chlorinated recesses of modern cities, bacteria have changed not because they seek to, but because conditions demand it. Their history is one of accommodation, invention, resilience, and their future, like their past, will be written in genes exchanged in plasmids, preserved in dormancy, and carried forward cell by cell into the yet unseen corners of Earth. And now, as the narrative softens into stillness, the long breath of bacterial time begins to fade into the background. No final moment marks their story because it never truly ends. From the earliest flickers of metabolic potential in ancient seas to the shifting lineages beneath our fingertips today, bacterial life has remained steady, unhurried, and continuous. There are no climaxes, no resolutions, only persistence, only change. In sediments, on skin, beneath glaciers, within clouds, their genes pass forward in silence. A strand of DNA recombines. A dormant spore stirs. A single cell divides once more. They do not seek to be seen. They do not ask to be named. Yet in the genomes of cyanobacteria and sulfur, oxidizers of symbionts and extremophils, the history of Earth is recorded not in chapters but in sequences. Their legacy is not only in oxygen or iron or decay, But in the quiet equilibrium of life continuing adapting and dispersing through every gradient the planet offers, they are the breath beneath our breath, the architects of possibility. So now the microscope dims, the tree recedes, and the soft threads of biofilm, the drifting filaments, the scattered spores, these fold gently back into the silence. Let them remain there a while beneath stone, beneath thought, beneath time.